So I've been talking about a lot of the research in our group that's focused on understanding how proteins in the cell will self-assemble with one another at the right place and the right time in order to control important functions in the cell, and we're especially motivated by trying to understand clathrin-mediated endocytosis, which is essential for the survival of all eukaryotic cells.
What is it?
So it's a pathway for transporting material across the cell plasma membrane. So there's receptors in the membrane, and they bind nutrients, and then those need to get inside the cell. So in order to do that, clathrin and about 60 other proteins actually assemble on the membrane and cause it to deform into a vesicle. So they drive the remodeling, and then that buds off the membrane and gets traffic inside. So iron, for example, is something that gets transported through clathrin-mediated endocytosis, but also the receptors themselves get recycled. And so it's important for controlling signaling.
Okay. And is it all simulation?
Yes, it's all simulation.
So how do you know that it's accurate?
So that's a great question. And luckily, it's such an important pathway in the cell that it has been studied for decades, and so there's quite a bit of experimental data. There's structural data on a lot of the proteins, there's biochemical data on their interactions, and there's in vivo data on the dynamics. So we can use that to help parameterize the model, but then because it's complex and involves so many components, we need to match our output to actual experimental observables. So we have several collaborators that can collect data with sort of very well-defined initial conditions, and then we can use our model to fit the data and actually predict what will happen when you change, for example, the concentrations of the proteins or the lipids that they bind to.